The cell, the basic building block of all living beings. And this is the nucleus of a cell. The nucleus contains all the genetic information pertaining to our bodies. Genetic information is stored in 23 pairs of chromosomes. Each chromosome contains a long DNA strand, which is packed tightly around proteins called histones. DNA contains gene segments, where the instructions needed to make proteins are kept. When a protein is needed in any of the approximately 100 trillion cells in the body, a signal is sent to the DNA via hormones. After that, an enzyme named RNA polymerase steps in to retrieve from DNA the information to be used for the protein. However, this is no easy feat, as finding the right code needed to produce a certain protein among the huge database of DNA is similar to finding a single page in a 1,000-volume encyclopedia. However, the enzymes very rapidly read the information in the DNA from the beginning to the end and find the code quickly without making any mistakes. Only the relevant part of the DNA will be unfolded. The enzymes will keep the relevant part of the strand unfolded, while the remaining sections remain in their folded form. RNA polymerase attaches itself to the starting point of this gene that contains the code and travels through the DNA and forms the messenger RNA using the bases that are freely circulating in the nucleus. The code in the DNA determines the order in which these free circulating bases are added to the messenger RNA. Messenger RNA now has a genuine copy of the necessary gene. Messenger RNA must go through a process before it can be used as a template for the protein production. In this process, some parts of the RNA are removed and spliced together. Now, it has received all the necessary information, no less, no more. With this important information, the messenger RNA is ready for the factory, that is, the ribosome, in which the proteins will be produced. The messenger RNA is now ready to leave the nucleus with its valuable cargo. However, there are gatekeepers waiting at the gate of the nucleus. They decide what goes into the nucleus and what doesn't, and open the gates accordingly. However, for the messenger RNA, the gates will always be opened. With the information it copied from the DNA, the messenger RNA goes straight to the ribosome and never gets lost. Ribosome is the factory that produces the new proteins. The messenger RNA binds to the relevant part of the ribosome, and preparations begin for the production of the new protein. The ribosome, in other words the factory, needs raw materials to start production based on the information brought in by the messenger RNA. These raw materials are amino acids, which can be in 20 different forms in living beings.
The ribosome knows what amino acids correspond to the letters sent by the DNA and allows only the correct amino acids to enter. It takes the ribosome only 4.5 nanoseconds, which is 4.5 billionth of a second, to identify an incoming amino acid. The technology and intelligence displayed by organelles, which are only 100,000th of a millimeter, is spectacular. Thinking or even imagining that these details could come about by chance is obviously very illogical.
Transfer RNA brings the raw materials and positions them according to the code. The instructions that contain the information for the production of the protein have arrived from the DNA. However, there's a problem that needs to be solved. The written instructions on the order slip are in the DNA, which is written in a special 4-letter alphabet. The language of the proteins to be produced is completely different and consists of 20 different amino acids. In other words, it has a 20-letter alphabet. The conversion of this language difference is called translation. Ribosome performs this translation between the two languages. Called the codon-anticodon method, this translation system works much better than the most advanced computer centers around the world today. It acts like a professional translator expert in both languages. The messenger RNA that previously arrived at the ribosome carrying the information for the protein production and the transfer RNA carrying the amino acid at its one end find each other like a key and a lock. Within a very short period of time, the information written in the four-letter alphabet of DNA is translated into protein language. Amino acids attached to the upper end of the transfer RNA form an astonishing protein chain, each consisting of at least 100 amino acids. In order for the translation to function in the best way possible, the ribosome enlists the help of over 100 assisting molecules, each of which works in concert with one another.
Protein synthesis is actually a translation process from one language to another. As you can see, our genetic information is translated from the language of DNA to the language of protein, and the translation is done by an organelle called a ribosome.
Here, the amino acids are linked by special chemical bonds called peptide bonds, which are among the strongest bonds. For this reason, proteins are sturdy and are not easily damaged by external effects. After the new protein chain is formed, the chain assumes its unique three-dimensional shape through a series of folding moves. This three-dimensional shape will determine the quality of the protein. This shape is so important that the protein would be useless if this unique shape was not fashioned, even if the sequence was correct. With the formation of the special three-dimensional shape, the process is now complete, and new protein is formed. When the new protein emerges from this special compartment, an enzyme on duty arrives at the scene. It picks up the resulting protein and takes it to its destination where it is needed.
When it comes to protein synthesis, you have what I would call the queen mother of all chicken and egg problems. Because in order for proteins to be made, you have to have proteins in place. Proteins are reading the information in DNA so that those instructions can be copied by the cell. Those instructions are carried by proteins to the ribosome. At the ribosome, which is made up of a large number of proteins, there are proteins that are assembling proteins themselves. And in fact, even when proteins begin to fold after their production, it's proteins that are helping the proteins to fold. So you can't have proteins without proteins, but the problem is even more severe for an evolutionary paradigm because if you are starting off with proteins that are less efficient that quote-unquote evolved to be more efficient, that scenario could never work. Why? Because if you have inefficient, less effective proteins functioning, they're going to be producing defective proteins, which means in turn the proteins that make proteins are going to become more defective. And when that happens, you have something that spirals out of control and is, in effect, a death spiral for protein synthesis. So there's no way to envision how something like protein synthesis could evolve because of the chicken and egg nature of that.
Right, so protein synthesis is actually a fairly complex series of events that occur within the cell at an incredibly fast rate. It involves multiple proteins that are necessary to transcribe the DNA into RNA, and then additional proteins that are required to translate the RNA into proteins. So protein synthesis actually requires both DNA, proteins, and RNA, as well as transfer RNAs and ribosomal RNAs, the latter of which also include different protein subunits. So it's quite impossible to get proteins without proteins already being in existence.
The process of protein synthesis, which we tried to summarize briefly, takes place 2,000 times every second in every cell. All the units involved in the process are very well aware what they need to do. Not even a single enzyme gets confused about its goal. There's never a faulty production in the factory. The unique folding shape of the new protein chain is never wrong. The carriers and messengers never lose their way. The cell is always aware of the importance and sensitivity of this special production. It is aware because Almighty God inspires every component inside the cell what to do. Each enzyme, each amino acid, every atom that makes them up is the manifestation of the supreme power, endless knowledge, and the superior intelligence of God. Everything submits to God, who is the owner and ruler of all the worlds, who is everywhere and surrounds everything at all times. Everything is in His knowledge, and everything has surrendered to Him. It is indeed very easy for all-merciful God to create all these amazing wonders out of nothing. It is not possible to explain such a complex system like protein synthesis with an evolutionary mechanism. Evolutionists are also aware that it is impossible for the protein molecule to come into existence by itself through a series of coincidences. The theory of evolution cannot explain how a single amino acid initially came into existence, let alone a protein molecule. Evolutionists have conducted many experiments in their attempts to explain the origin of life. The Miller-Urey experiment, which was conducted in 1953 and is their most respected study, is one of those efforts. With the experiment, these two biochemists hope to be able to discover a finding that could suggest that the amino acids, which are the building blocks of proteins, could have coincidentally formed in the lifeless world billions of years ago. However, the experiment did not correctly mimic the atmospheric conditions and required plenty of external intervention. The result was the right-handed amino acids. However, organisms do not use right-handed amino acids. Nevertheless, the experiment revealed one truth: amino acids could be obtained only in a laboratory environment, the conditions of which are specially arranged and through intentional intervention. Even the smallest protein molecule boasts incredible features, fascinating mechanisms and properties. It is impossible for lifeless atoms to organize themselves to form such perfect structures through coincidences. In other words, the power that gave rise to life was not unconscious coincidences, it was the creation